Namaskar. In this video lecture, I will talk about ubiquitination. Ubiquitination is a biochemical process. It occurs in all cells and it's a process to tag proteins which are destined for degradation. Misfolded proteins or toxic proteins or proteins which have completed their functions and they are no more required in the cell have to be degraded. Otherwise, they will accumulate in the cell and will cause various kinds of problems. So this degradation, how those proteins will be identified, how the system will recognize that this is the protein which has to be degraded. So that mechanism is called ubiquitination. In this process, several small proteins called ubiquitins, 76 amino acid long proteins, say I'm indicating that protein like this, a question mark like this, laterally oriented. So this is ubiquitin, say. So it is a 76 amino acid long polypeptide. Probably it is universally present in all eukaryotes, and there are also prokaryotic version, and hence it is called ubiquitin, ubiquitously present. So these ubiquitin proteins, polypeptides, will be tagged to a target protein. Say this is the target protein. Say this is the target protein. So this target protein is a misfolded protein or it has completed its function. You have heard about ubiquitination several times in cell cycle. The cyclin proteins are cyclically produced and degraded by a proteasomal degradation. So before going to proteasome, this kind of proteins, this one, like for example, will be tagged by these ubiquitin proteins. So it will be tagged at its lysine subunit. The epsilon amino group of the lysine residue will be tagged by these proteins. Say several of these ubiquitin molecules. So this is called polyubiquitination. So they will be tagged by several such molecules with the help of a ubiquitin ligase enzyme. We will talk about the enzymes also, how that occurs. So when this is being tagged by a chain of ubiquitins, this protein will go for degradation in the proteasome. Proteasome is a protein degradation machine. It comprises of 2 alpha helix and 2 beta sheets. The beta sheets are sandwiched between the alpha helices. So this is alpha, these two are beta, and this is alpha layer again. So it's a kind of a pipe-like protein, in fact, and this comprises the 20S sedimentation coefficient, and it has a cap protein, that is 19S cap, and on the other side also there is a cap, 19S cap, and this it may be so that there are proteasomes with one cap only. So this completes the 26S proteasome, which is the completely functional proteasome. So this protein will now pass through this, the ubiquitins will be identified by this cap-like structure, 19S cap, and it will push the protein into this like this. So the ubiquitins were there like this, and they have been actually identified by this cap-like 19S cap, and the protein is pushed into the proteasome. And when it comes out from this, it is degraded into small peptides, which can further be degraded into amino acids so that they can be used in the next round of protein synthesis. And the ubiquitin molecules are released. The ubiquitin molecules are depolymerized after that. So this is what happens with proteins which are in the ubiquitination process. Now, this tagging of ubiquitin, it involves several enzymes. There are 3 enzymes, in fact, they consecutively act: E1, E2, and E3. E1 is an activating ubiquitin activating enzyme. E2 is a conjugating enzyme, and E3 is ligase. It's a ligating enzyme. It will ligate the ubiquitin residues to the target protein. So it occurs like this. First, the E1 says this is the ubiquitin. Ubiquitin has a carboxyl group. So this is the carboxyl group, say. So it is tagged with the E1 protein first, with the investment of an ATP residue. ATP molecule is invested, and the ubiquitin forms a thioester bond with the E1 cysteine residue of E1 enzyme. So the E1 is now bonded with, if I can draw it like this, so the sulfur is on this side, and one water molecule will be removed and this thioester bond will be created. So it looks like this. So now this ubiquitin will be further transferred to the E2 enzyme, which is the conjugating enzyme. So it will take the position of E2 with this cysteine residue, again with the help of a thioester bond. The ubiquitin will now be tagged to the E2 enzyme. And finally, this ubiquitin will be transferred to the target protein with the help of the E3, which is basically ligase, ubiquitin ligase. It transfers the ubiquitin to the target protein like this. And this will occur continuously to tag several molecules of that to complete the process. So this protein is now stamped or targeted or tagged with a kind of a signal that will be identified by the proteasomal complex for degradation. So this is the function of ubiquitination, but there are several other functions that are coming up recently of ubiquitination. Ubiquitination might occur once or multiple times with the target protein. It may be mono-ubiquitination or poly-ubiquitination or multi-ubiquitination like this. Suppose in case of mono there will be one ubiquitination. There in case of multiple or multi-ubiquitination there will be multiple ubiquitination at different sites. But poly-ubiquitination, it occurs on the ubiquitin molecules like this, one after another, like a chain. So this is for degradation purpose, but these two may have different roles. So some of the roles of ubiquitin are, one of the role is in histone code. So a kind of epigenetic regulation in which the ubiquitins are tagged to the histone residue H2A, H2B, H3 and H4. So due to the covalent addition of these ubiquitins to these histone proteins, the expression of the genes changes. This is called histone code, one of the type of epigenetic regulation. In other case, the ubiquitin might be involved in signaling or trafficking, protein trafficking through membrane. Or it may be in one of the recent finding is this: the list is increasing, the role of ubiquitination is increasing. There is one kind of you can say repair mechanism or escaping DNA lesion in the DNA by using ubiquitination. So what happens is during replication in eukaryotic system, suppose here is a lesion and the DNA polymerase has halted here. It is halting here, and because this it is not able to recognize this lesion and to cross the lesion. But there are mechanisms that the ubiquitination mechanism which will help this or the other polymerase, alternate polymerase are there, which will overcome this lesion. So this is a kind of a, you can say, post-translational modification. There is a PCNA sliding clamp with the polymerase, DNA polymerase in eukaryotic system. So that will be ubiquitinated. That will get ubiquitinated, and when it will be ubiquitinized, what will happen? It will recruit another DNA polymerase, which is the family of UMUC2 family of DNA polymerase. So that DNA polymerase is lesion. It can overcome the lesion. It can replicate, even it will replace this DNA polymerase by itself when it will recognize that this PCNA or sliding clamp has been tagged with ubiquitin. So it will replace it, and it will replicate this lesion also. So it's a kind of overcoming or escaping DNA lesions. So this is how ubiquitination is involved in proteasomal degradation of proteins which have completed their functions, like the cyclin proteins, or the protein we were talking, like the securin protein. In the last video we were talking about during anaphase, the securin is degraded by ubiquitination to release the separin, which will cleave the cohesin proteins that have bounded the chromatids to separate them, and then the cell will enter into the anaphase. So this was all about ubiquitination in this video. Thanks for watching. I would at least like you to press the like button, at least, that I will understand, okay, somebody is liking it, and I will prepare more videos